Hi students, it's always my pleasure to talk about the important aspects of your chapters from biology for class 11 CBSE students especially, because here you have to know the portions in a little detail. So in this chapter, that is the body fluids and circulation, the remaining parts I have to discuss here in this video. So blood is the main fluid which is being circulated, and about blood you have heard it a number of times and you've read also. So the important aspects, the features you have to remember, like composition, that is plasma is there. 55% is plasma and 45% is formed cells. So this is the composition. Plasma again containing 91 to 92% water, right? And 7 to 8% proteins, and your 1 to 2% inorganic salts, right? So plasma is the liquid part of the blood, and the proteins that it contains are very important, like your albumin, which controls the blood pressure, and then the immunoglobulin, which enhances the body defense system or immunity, and then the fibrinogen, which is helping in the blood clotting process. You all remember, like, right? Then this is the plasma content of the blood, which is actually the liquid part of the blood, and it's containing a number of substances, important substances, and also some waste are also there in the plasma, which are being excreted through the kidney. So everything which is there in the blood, including the waste, are there in the plasma, right? So this is about plasma, you have to remember. And about the cells, RBC, WBC, that is erythrocytes, leukocytes, and platelets, that is thrombocytes. So about your RBC, erythrocyte, it is the smallest cell of the body. It does not have nucleus. It is having hemoglobin, that is containing iron. That is why the blood is red. Hemoglobin is the main respiratory pigment in mammals and in human. That is why blood is red. In some lower animals, the blood is not red because hemoglobin is not there. The other pigment, which is hemocyanin, is there, which is containing copper, and that is where their blood is blue, like lobsters, crab, etc. Okay. RBC lifespan 120 days. Shape biconcave shape. Nucleus or no nucleus. Always remember, there is no nucleus in the RBC. Why? Because it has to accommodate and carry oxygen only to all the body parts. So it does not require any other organelle. No mitochondria, no endoplasmic reticulum. Okay? So this is the absence of these organelles are giving extra space for the RBC to accommodate and carry oxygen, which is its main function, in the form of oxyhemoglobin to all body cells and tissues, where oxygen is released, and also taking back carbon dioxide in the form of carbaminohemoglobin after combining with hemoglobin. So carbon dioxide is also taken back and is released through the exhalation process in breathing. So this is your RBC, okay? And the main features of RBC you have to remember, okay? Was formed in bone marrow and is destroyed in liver or spleen. So normal count of RBC is about what is the normal count of RBC? It is about 4 to 5 million per cubic millimeter of blood. So that is the 5 millions is the normal count. 5 millions to 5 millions to 5.5 millions per cubic millimeter of blood. This is the normal count, okay? And They have a red color, iron containing complex protein called hemoglobin that I have discussed just now. And about leukocytes, the normal count is not much. It is 4 to 5,000 per cubic millimeter blood. Okay, or you can say it can average. The normal count can average from 6,000 to 8,000 millimeter cubic millimeter of blood. So this is the normal count that you should remember. Okay, if there is any question like this related to the normal count. Okay, now the shape of WBC, it is amoeba-like shape, irregular shape, having pseudopodia, finger-like projections by which it engulfs the invading bacteria or any other pathogen. Okay, it is having nucleus, right? And the types of WBC you have to remember. There are two types. One is granular leukocytes or granuloleucocytes, and the other is agranular or non-granular leukocytes. So granular leukocytes consists of your neutrophil, eosinophil, and basophil, and your granular or non-granular leukocytes consists of your lymphocytes and monocytes. So the bulk of WBC is actually the neutrophils, which are about 70% in the total WBC. So neutrophils actually do the phagocytosis, that is engulfing the bacteria or any other pathogen. The engulfing process is known as phagocytosis, which is done by the neutrophils mainly. And in the non-granular part, the lymphocyte is the most important ingredient, lymphocytes, which are a special type of WBCs, and they not only kill the pathogen but also form antibodies against a specific pathogen. So there are two types of lymphocytes. One is T lymphocyte and another is B lymphocyte. T lymphocytes are mainly collected in the thymus. So that is why they are called the T lymphocytes. Okay, and which is there in the lymphatic system, thymus, a part of the body. So that there these lymphocytes are collected or they assemble there. So that is why they are called T lymphocytes. And B lymphocytes, actually the B name has been derived from the bursa, which is a part of a bird. So that is why it is called B lymphocytes. But these lymphocytes in totality are the very strong, WBCs, which not only kill the pathogens, engulf the pathogens, but also form antibodies. So these T cells, especially, they kill the bacteria or the pathogens, and B cells, they form the antibodies also. So that there are two types of lymphocytes, T cells and B cells. So these are the agranular WBCs or leukocytes. So you have to remember the names and in the both the types of WBC. Okay, so lifespan of WBC is from few hours to 3 weeks, or few hours to some days, and it is having nucleus. Okay, and it is formed in bone marrow and killed while fighting with the bacteria. In the pus also it is formed, dead WBCs. Okay, and then the platelets, that is the smallest, that is also a very small blood cell, and it helps in the blood clotting process. Okay, it also does not have any nucleus. So these platelets, how they help in the blood clotting process? Actually, they release a chemical which is known as the thromboplastin, and this thromboplastin actually convert the thromboplastin converts the prothrombin into thrombin in the presence of vitamin K, which is a fat-soluble vitamin. And this thrombin further converts fibrinogen into fibrin, which is actually the clot. So, in the presence of calcium ions, so these are the vitamin K and calcium ions help in the conversion of these proteins. So, firstly, it is the thromboplastin, which is released by the WBCs, which accumulate at the site of the wound. And this thromboplastin actually is doing what? It is converting the prothrombin into thrombin in the presence of vitamin K, which is a fat-soluble vitamin. And then also this thrombin is doing what? It is converting the fibrinogen into fibrin, which is the final clot. Okay, so this mass, solid mass, it forms the clot and the bleeding is stopped. So this blood clotting process also you have to remember, students. So this is all about the blood cells. So this is about the composition of the blood and the details of these ingredients. Okay, so composition of blood, main parts you have to remember here. And about the blood grouping, Carl Landsteiner actually came out with the process which is known as ABO process, and this is how the blood is grouped. Because if there is an emergency, you have to donate blood to some needy patient, then the blood group has to match. Otherwise, there will be a clubbing of blood cells in the recipient, and this can be very dangerous and can be fatal also. So, if the blood group of the donor is not matching with the blood group of the recipient, then what will happen? The blood cells will club together and will not allow the blood to flow. So this is a very, very dangerous situation. That is why the compatibility of the blood has to be matched of the donor and the recipient. So what he did was he studied and came out with the theories that the blood of human blood, every RBC, in the RBC there is a protein, there is an antigen, okay, and in the plasma there is a antibody. So if the antigen is A, then the blood group is A, and if the antigen is B, then the blood group is B. So if the antigen is A, the antibody will be B. If the antigen is B, the antibody will be A. So like this it goes, and then he was able to find out the compatibility chart of the different groups. So he came out with 4 groups: Group A, Group B, Group AB, and Group O. Group A means, as I said, the antigen in the RBC is what? Is A. And in the group B, the antigen is B. And the group AB, both the antigens are there. Group AB contains both the antigens A and B. And group O does not contain any antigen, but it contains both the antibodies. So likewise, what happened is that the compatibility was studied, and then it was found that which blood group can be given to which blood group. So the matching was found. Similarly, group A can be given to group A only and group AB, okay, and group B can be given to group B and group AB, because antigen A and antigen A is there in both, in group A and also group AB. So there will not be any antibody antigen reaction here. Similarly, AB antigens on RBC is AB, okay. So AB can be given to AB only. So like this, the matching was found. O is universal donor, can give blood to all the other groups because it is not having any antigen. So there will be no chance of antigen antibody reaction. But it can take blood only from its own group. So it is a universal donor and it can take blood only from its own group, that is O group. And AB is an universal recipient. It can take blood from any group because both the antigens are there. Okay. So it is an universal recipient. So like this, matching and compatibility of blood was found, how it can be done, and the onus goes to Carl Landsteiner. Okay. So donor compatibility is there in your chart, in the chart in your book also. Blood group A, B, AB, O. A, the antigens on RBC is A, antibodies in plasma is anti-B. Donors group can be A and O. Can be A and O. O is the universal donor. And if the blood group is B, antigen on RBC is B. So it cannot be given to A blood group, can be given to B and O. B is its own group and O is the universal donor, so it can be given to O also. AB, in the blood group AB, the antigen on RBC is A and B both. So, and there is no antibody in the plasma. So it can be given to AB, A group, B group and O group. So it is it can take blood from all these blood groups. So it is an universal recipient. And O, there is no antigen, so can give blood to all the groups and can take blood only from its own group. So this is clear from this chart, and I think this will help you in remembering and understanding the blood grouping. RH grouping also there from the rhesus factor, the monkey. So everybody commonly is having this RH factor in the blood, but if there is no RH factor, then it is RH negative. So all the blood groups which are having RH factor is RH positive, and those who are not having this factor is RH negative, right? So there has to be compatibility in this also. The RH negative cannot be given to RH positive. So this matching has also to be done. But in during pregnancy there can be a problem in the second child, the birth of the second child, or in the during the second pregnancy, if the mother's blood is negative, like O negative, and if the father's blood is positive, any group, because O can take blood from any other group. In the first child, in the case of first child, there will be no problem, but in the second delivery, there can be some problem, because by this time the antigen antibody reaction can start in the mother's womb. So the development of fetus can be damaged, can be hampered. So there one has to take precautionary steps, and there are injections which are given after the first delivery, which neutralizes this antigen antibody reaction at the time of this second pregnancy, right? So this you have to remember about the RH factors. Okay. And about the circulatory system, I have already discussed. You have to remember that in human it is double circulation always. So the deoxygenated blood is going to heart through which vessels? Through veins. Through which part of the heart? That is right auricle. And when it is a common diastole in the auricles. So the right auricle is filled with deoxygenated blood. Then what will happen? The tricuspid valve, which is there between the right auricle and right ventricle, will open by the pressure of this blood, and the blood will enter to the right ventricle, right? When it was in the common diastole of the ventricles were there, the ventricles received this blood, deoxygenated blood, from the systole when from the auricles when systole is there. When auricles, the contraction in the auricles were there, the blood entered the right ventricle. Okay, now right ventricle will contract, systole will happen or take place in right ventricle. So this right ventricle, from this right ventricle, the deoxygenated blood will go to the lungs. Through which vessel? Through pulmonary artery, the only artery which is carrying deoxygenated blood, right? So why it has gone to lungs? Because here the gases exchange will take place, and this deoxygenated blood will become oxygenated in the alveoli. So this oxygenated blood will be carried back to the left auricle of the heart in their common diastole. So this by which vessel? By pulmonary vein, the only vein which is carrying your oxygenated blood. Now from this left auricle, the blood will be pumped to the left ventricle. So through which valve? Through the bicuspid valve. Okay, now in this left ventricle, systole will take place, contraction will take place. When it was in diastole, it was filled by blood, by this oxygenated blood. But now it will contract in the systole, and the blood will flow through the semilunar valves to all parts of the body. So there are semilunar valves also. One is here at the when the blood is passing out from the left ventricle to all parts of the body, and also at the point where the deoxygenated blood is going from the right ventricle to the lungs. There are also semilunar valves are there. So the sound of love dove is actually the closing of these valves. The sound of the closing of these valves. When the tricuspid and bicuspid valves are closing, it is love, and when the semilunar valves are closing, it is dove. So this is the love and dove sound, and the circulation has taken place. The complete cycle, has taken place, which is called the double circulation. Once through the lungs, so from the heart to the lungs, lungs to heart. So this is the pulmonary part, okay, and the other part is from heart to body and body to heart. That is the systemic part. So that is why it is said that in human the circulation is double circulation. Okay, first is going to the lungs and then coming back again after the blood has become oxygenated to the heart. So the blood which has become oxygenated will now go to the other body parts and will come back again after releasing oxygen to all the cells. So this deoxygenated blood will come back to the right auricle again. So this is the heart from heart to body, body to heart. So this is the other part, which is the systemic part. So that is why it is called the double circulation of heart. Students, very clearly remember that this is the double circulation, how the blood is entering the heart and how it is going to the lungs and then again coming back to heart and then going to the other body parts after becoming oxygenated, right? So I think this is covered almost all the important points of this chapter, and I end my video here and will come back with other important parts of your chapters of class 11. So thank you very much. God bless you all.